In this presentation, I am very happy to share with you summary of interview feedback from industry stakeholders. As you know, communication is a central mission of CRCG. Through this communication, we hope to establish a neutral channel to facilitate interactions between industry and the FDA. Use this channel to outreach to industry to understand what are the main pain points in development and approval of complex generic products. We also hope to use this channel to solicit feedback from the industry stakeholders on product-specific guidances and other guidances that had been developed by the agencies. We plan to publish our research and peer review publications and use to assess the metrics of CRCG success. Prior to the interview, we sent an email asking several fairly open-ended questions. We wanted to understand what are the main barriers for development of complex generics and what are the industry pain points around those products. Are those barriers due to scientific issues, regulatory challenges, market competition, or ROI considerations? We also asked about how the center could help the development of complex generics through either research, training, or facilitating communication with the agency. Some interviews were very well structured and the company came prepared with slides and written summaries, and some were not, and all of the interviews allowed for an open dialogue. This information will be used to focus the efforts of the center as well as to help FDA's ongoing GDUFA-funded activities. We plan to publish the outcomes of this interview in a peer-reviewed publication. In total, we interviewed 20 organizations, including some of those interviews were conducted multiple times. The type of products discussed included complex injectables, liposome, long-acting injectables, iron colloids, inhalation, topical and transdermal products, ophthalmic, emulsions and suspensions, and orals. The main issues brought up by the companies included CMC issues for either specific products or product classes, old and variable products, bioequivalence issues, clinical trial design, use of modeling and simulation, product-specific guidances and the changes of product-specific guidances, general communication concerns, leachables and extractables, and nitrosamines. Here we summarize the companies we reach out to. Those include large multinational generic companies like Perrigo, Apothec, Teva, Viatris, Sandoz, Cipla, Amneal, and Sun Pharma, as well as the smaller specialty companies such as Fresenius Kabi, Cassette, Solaris Pharma, Nexus, Xillia, Capstone, as well as trade organizations. We also highlight the type of products that have been discussed during the interviews. I would like to use this opportunity to let you all know that if you are not on this list, please reach out to the center and we will be happy to talk to you, as well as the fact that we would like to continue those interactions on an ongoing basis and perhaps interview you again in the next 6 months as the field is constantly changing. As you can see by this table, the number one concern brought up through those interviews were specific issues with chemistry, manufacturing, and controls. Those issues were related to either specific products or broadly to general product categories. They also included drug-device combinations, as well as important issues around old and highly variable products. Companies also discussed specific bioequivalence and clinical study design issues, as well as changes in product-specific guidances, leachable and extractables, and nitrosamines. The first item I would like to discuss is the use of modeling and simulation to supplement bioequivalence studies. It is absolutely clear that modeling could help development of complex generic products. The number one product category brought up when modeling was discussed in those interviews were utilization of PK/PD modeling for inhalation products. It's also as well as for oral dosage forms. For products like ophthalmic, topical, and oral inhalations, the in silico modeling is very helpful. It's also generally agreed upon that populational pharmacokinetics to discuss the differences of the site of absorption is very useful, and modeling around long-acting injectable studies also could be beneficial. However, industry had a number of major concerns. They all understand that it's important finding alternatives for performing of expensive clinical endpoint studies, but no single company would like to be a trailblazer in this area. There are also kind of lack of general models that are accepted by the industry and the agency for complex generics. What we realize that GADUFA research sponsored development of multiple models, but also the companies themselves either use software or have their own software written to predict modeling of deposition of inhalation products, for example, and the impact on pharmacokinetics and bioequivalence. But there is not a single model that is acceptable for everybody and agreed upon by the agencies. They also think there is kind of general lack of publications on what are the expectations for the bioequivalence modeling and what would be required to validate the model. Everybody believes that they could really benefit from a FDA feedback during model development, and companies would really like to have meetings to discuss their approaches as they develop their models. While modeling is often used, nobody knows how modeling will be reviewed by the agency and whether some of those modeling approaches will be accepted. And this increases regulatory uncertainty around the use of modeling for generic products. There was very specific questions. For example, for cytotoxic drugs, many times some of the time points are missing, and companies were wondering if they could use modeling or partial modeling to supplement some of this patient data. About 60% of our time during the interviews were devoted for different chemistry and manufacturing control concerns. The number one concern is development and validation and the lack of standard analytical methodologies for physicochemical characterization of complex generics. I think as an industry, we all aware how to establish and validate a typical HPLC method. However, anytime we talking about characterization of complex products, there are no standard methodologies out there. For example, it's not clear how to measure particle size and morphology, how to conduct in vitro drug release testing, how to assess peptide immunogenicity, patch adhesiveness. There are also no clear guidelines of how to validate this method. Some of those methods very difficult to validate. It's also not clear how best to show their discriminative ability. Many of those methods really are highly variable and therefore cannot be held to the same standard as typical HPLC type of assays. There are another issues around analytics that was brought up by several companies that they develop more novel, discriminative or highly sensitive methodologies that well accepted within the industry, but when they submit those results with filings, often they are not accepted by the agency because they do not match USP methodology that many times not as sensitive or not as accurate. There are also a number of so-called boutique methodologies that are brought up by the agency in their publications, and the companies don't have access to this fancy equipment and do not know if it's expected for them to use those methods to characterize their products. Those are morphologically directed Raman spectroscopy, various imaging techniques, and small angle X-ray diffraction methods. The next issue brought up was it's a very extensive analytical requirements discussed in product-specific guidances. Several companies said it pretty much this way. They said there are so many parameters listed as important in the guidance. Are all of them equally important? Do we have to measure just some of them, or are we required to measure all of them? And they have the thinking that many of those parameters really do not impact bioequivalence, and yet they have to measure and match them. Some of those requirements appear to be almost of purely academic interest, and there are no specific scientific rationale or data that supports that this particular requirement is important for product bioequivalency. Some of the examples were hydration layer for the liposomes and particle drug distribution within the pores of long-acting injectable microsphere products. Other general CMC issues included: do you have to be Q1, Q2, or can you be Q1, Q2 like? In many instances, company believes that being not precisely Q1, Q2 within 5% of variability still results in products that are bioequivalent. And in many instances, there are some patent considerations that box the companies out from achieving the complete Q1, Q2. But there are no alternatives of how to use if Q1, Q2 is not fully achieved. In many instances, product-specific guidances are so detailed that Q1, Q2 route appears to be almost more complex than clinical endpoint studies. It's also difficult to achieve Q1, Q2 equivalence for products that have over 10 ingredients. Specifically, companies brought up many topical products, including petrolatum and waxes, and difficulty in figuring out exactly what excipients are there and matching Q1, Q2 for the specific excipients that are only 1% in the formulation. In addition, companies mentioned there are some specific very small issues in Q1, Q2, like a buffer composition or osmolality that holding off product approval. In addition, there are some products where clearly there is a difference between the label provided by innovator company and the data reverse engineering that was achieved by the company. Other general CMC issues included RLD, API, and excipients availability. It is difficult to procure 3 lots for some of the drugs, especially drugs with low volume, orphan drugs, or drugs that are very highly priced. There is a requirement that you have to have N equals 3 of everything, and for some of those RLDs, only 1 batch is produced per year. Some of the excipients, only 1 batch is produced per year, which means that the company has to collect batches for 3 years before the start of reverse engineering and bioequivalence. And some RLDs are just no longer available. The next issue, very important, is around highly variable or old RLD products. There are several products, and they appear to be very important products with high markets and no available generics. They themselves are highly variable. If you buy 2 lots of the RLD, many times they wouldn't pass bioequivalency or in vitro comparability when compared to itself, based on drug release rate, particle size, or purity. When bioequivalency studies are conducted using 2 different lots of the products, again, many times variability is greater than the specifications set by the agency on bioequivalence. The products that were mentioned in this respect were iron sucrose, PLG, long-acting injectables, dose-metered inhalers, and topical steroids. Additionally, there are clearly some discrepancies between label and actual product, such as drug overages, which make it very difficult to develop generic products and file. For some of the products, there are lack of availability of API and excipients, especially for some of the old products, and again, some of those batches made only once per year, making it difficult to collect 3. Additionally, some of the old products, and in a way easy to manufacture, they produce using very old technologies, Examples were given like estradiol creams and other topical steroids that could, the particle size lot to lot is so variable. The company would like to use more novel methodology to control the particle size, but then they do not match the variability of the initial product. In some instances, the formulations are not very stable, and the company would like to add a small amount of antioxidants to stabilize the formulation, but then the formulation will not match Q1, Q2 requirements. This slide highlights some of the most frequently discussed products during the interviews. Liposomal products, long-acting injectables, and iron sucrose and ophthalmic emulsions were brought up by a number of companies. For liposomal products, there is difficulty in development and validation of some standard analytical methods to characterize particle size, particle morphology, in vitro drug release, lipid impurities. There are also many parameters required to be measured by product-specific guidances, and many times the company already completed bioequivalence study and they have to go back and measure many parameters. There are also sometimes products are morphologically different, but they behave identically in vivo. For long-acting injectable products such as PLGA, similarly, many methods were highlighted, such as morphology, porosity, polymer purity and particle size, in vitro drug release, and also drug distribution within the microspheres. Characterization of peptide aggregates within the microspheres and potential impurities impacting immunogenicity in complete drug release were mentioned by several companies, as well as RLD variability. Iron sucrose was a big topic with difficulty in methods and drug variability, and also ophthalmic emulsions. The same, difficulty in characterization, variability of RLD, and also it appeared to be some small Q1, Q2 issues preventing approvals. Inhalation products are very unique. It is like a combination of the entire pharmaceutical science field in one type of drug. They're also very important product for the industry, and many companies are working on dose-metered inhalers because those are big products and there are very few generic versions available. Yet they represent very unique challenges, especially for poorly soluble in drugs and dose-metered inhalers. There appears to be substantial batch-to-batch variability in the innovator products between particle size and morphology. The product also requires drug-device combination, and therefore there are certain patent considerations around the design of device and how the patient end up using generic device versus innovative device. There are clearly big differences in how patients use the device and populational difference in pharmacokinetics and response. Those type of products require very large clinical studies. People were estimating it's about $30 million studies to show clinical bioequivalence, and therefore companies really would like to have the ability to discuss with the agency prior to the start of the studies. Another issue with the entire development and clinical trials and development of dose-metered inhalers that there appear to be difference how they're regulated in European Union versus in United States. Those products require development of standardized method and standardized models, methods for defining particle size morphology, development of physiologically relevant dissolution methodology, as well as extensive modeling needs around pharmacokinetics, PBPK, as well as populational pharmacokinetics. Topical products were brought up by a number of companies. In general, industry is very appreciative of new guidance on IVPT studies that could be used instead of bioequivalence studies. However, there are a lot of outstanding questions around the design and conduct of those IVPT studies, and some of the statistical considerations that are not covered by the current guidance, and how to deal with the outliers, what to do, how to assess the impact of crystal formation, how to assess the impact of permeation enhancers. It's difficult to meet some of the specifications due to high variability, difficult to find n equals 3 of excipients and lots, and difficult to analyze the products that have many ingredients, over 10 ingredients. It's very difficult to establish Q1, Q2, when some of the excipients are only at 1%. The next couple of slides are focused on the clinical trial design issues. Several companies brought up the design for large endpoint trials for inhalation products, and their strong desire to look into alternatives for those endpoint studies based on PBPK modeling and populational PK modeling. Around the standard trial design and duration for bioequivalent studies, companies discuss long-acting injectables just because those trials are very lengthy, hard to recruit patients, and they would like to use alternative modeling-based approaches to shorten the duration of those trials or reduction the number of patients. They also want to look into the possible alternative for bioequivalent studies based on IVIVC or in vitro characterization. There is a limited number of patient pool, especially drug-naive patients for cytotoxic drugs. Another category discussed was poorly active topical products that are not Q1, Q2 equivalent, and how those studies large in variables and many times more expensive to prove equivalency of generics than approve the RLD. There is a very strong desire within generic industry to strive toward harmonization of clinical trial requirements between FDA and EMA and other regulatory bodies. This could really reduce the cost of clinical development of the products. There are issues around RLD sourcing for low-volume drugs, orphan drugs, or no longer marketed products. There is the need to source products specific for geography, such as Europe versus US, and in many instances the product is exactly the same and produced in the same plant. For some trials, the products expire before the clinical trial completes. There are other bioanalytical concerns around how to measure free versus encapsulated drug and plasma for liposomal products, or how you measure free or bound iron for iron colloids. And companies have examples of multiple bioequivalent studies failed due to product variability or the lack of robust analytical methods. For LAR products, discussions were around site of injection versus bioequivalence, and as well as correlation between adhesion and bioequivalence for transdermal products. Leachable and extractables is an important issue that generic industry faces that came up in a number of interviews. The main issue is the lack of consistency in how applications are reviewed. One company gave us an example of the same product submitted either by the manufacturer or by the distributor, absolutely identical end application, and one comes back with no deficiency and another has pages and pages of leachable and extractable deficiencies. Those inconsistency of the review in leachable and extractable cause a lot of delays in product approvals. Many companies see 9 out of 10 programs, so 80% of ANDA delays due to leachable and extractables issues. There is a need to repeat expensive studies, decreasing commercial viability of the products. And even low-risk products had to go back to generate additional leachable and extractable data. The company is sometimes unclear on the FDA expectations, and they would like to have an opportunity to discuss with the agencies their data and their deficiencies. They also see the same number of scrutiny applied, for example, for the cream for container, whereas the cream is stored for over a year, and for the applicator that sees the cream only for 1 or 2 minutes. This slide summarizes general communication and guidance concerns. Everybody loves pre-ANDA meetings. Companies find them very useful, and they would like to have another useful meeting in mid-review or post-CRLT where they could be able to engage in dialogue with the agency to defend their position or their application and to actually receive detailed feedback through this communication. In general, there is a desire for improved communication and feedback on expensive trial design or design of human factor studies or leachable and extractable studies. Another concern is companies would like to receive all the deficiencies during the first review cycle. Many mention that they submit their answers to the deficiencies only to receive new set of deficiencies during the second review that were not previously mentioned. Missed GDUFA goal dates due to complex review issues also appear to be concern for select products. Companies love product-specific guidances. However, they have the strong desire to be involved in product-specific guidance development, or at least understand where FDA coming from in the development of the guidance and provide the feedback. The changes in product-specific guidance appear to be a major problem for the industry. Many times, requirements are updated and the company have to go back to redevelop the product or repeat the clinical study, which really hinders economical viability of select products. It's also unclear to what standards the application already held to that had been filed, and also changes in the guidance means that later products held to a higher standards than earlier generics. How the center could help the industry addressing those concerns? We could perform reverse engineering for some of those highly variable products that are of high interest to the industry. We could establish and characterize performance of non-standard analytical methods, including establishing boutique characterization methods and making them available. We can understand the impact of Q1, Q2-like or morphological different products on bioequivalence, develop libraries of RLDS, APIs and critical excipients and make them available to the industry, as well as work with the agency on development of analytical methods for upcoming new product classes, such as RNA therapeutics. How can we address modeling and clinical trial concerns? I think it's important to work together with industry stakeholders and the agency to develop so-called standard models for inhalation, ophthalmic, long-acting injectables, and topical products. We could also jointly develop an understanding of what industry requires for model validation. In collaboration with industry, we also would like to explore alternatives to bioequivalence studies based on in vitro characterization and IVIVC, again for select high-profile products, and re-examine bioequivalence requirements for those highly variable RLDs. We also would like to attempt to work with FDA and other regulatory agencies on harmonization of clinical trial requirements. Additional concerns including working with the agencies to address leachable and extractable review issues, which appear to be an important problem concerning both new drugs and generic products, as well as develop analytical characterization methods and risk assessment for different nitrosamines, organize workshops on specific complex products or specific newly developed regulatory guidances to both allow for the agency to provide clarifications and also to allow for the industry to provide the feedback on how those guidances affect them. Also, we hope to be able to foresee potential scientific questions for products with patent expiries in the next 5 to 10 years, as industry is already working on those products. This brings me to a final acknowledgement slide, and first of all, I would like to thank the agency for awarding to us this U18 grant from GDUFA funding. I also would like to acknowledge multiple generic companies that agreed to be interviewed by the center. We're really grateful to Dr. Sam Rainey, the FDA grant manager for this grant. Sam not only helped us to set up those interviews, he participated in many of them, and he helped us to shape the vision for the center. Dr. Lisa Park from AAM helped to facilitate some of the interviews. I will also like to acknowledge Dr. Jim Polly from University of Maryland, the center co-director, Dr. Visha Krishnan, the center manager, and my graduate student Jill Coughlin.